now
Yes,
with
I
our
see the live button.
Google Plus Micro Journal Club. So Karen, take it away.
Hi, everyone. My name is Karen. I'm a PhD student at Simon Fraser University, and this is our Google Plus Microbiology Journal Club. And so Laura, my co-leader, and I, we
Hello.
wanted to, yeah, there's Laura. We wanted to get together and just gather a group of interested microbiologists, and we have all these awesome people that we meet on Twitter, and we wanted to talk about recent science papers. And so we have this very interesting paper that we're discussing today called New Antibiotic Kills Pathogens Without Detectable Resistance. And we do this regularly. We pick a Nature paper or some famous paper that everybody's talking about, and we talk about it. And what we want to do is we want to extend the invitation to everyone. As you can see today, we only have 3 people. Laura, Mark, and I have been getting together since the beginning of these journal clubs when we started last year. But we want to invite any new professors or students, PhD students, graduate students, master students, anybody who wants to join us to chat and you don't have to worry that you don't have anything to offer. I've just started these things myself, and everyone has fantastic things to chat about. And if you just come, it's a learning experience because you get to absorb all this awesome information and knowledge that everyone has to offer. And I always learn everything. I always learn a lot every time I'm here. And so I'm going to attempt to facilitate today, and that will be it. So you can see that we're here. I'm not a perfect graduate student, but we come and we want to talk about awesome science. And so we're just going to start out
Seconded.
today.
Yay. Sorry. I just wanted to yay. That was a good.
Thank you.
May I make a comment really quick? May I speak for the entropically challenged professors like myself? That it's really, really good to be involved anyway. One of the problems at a small liberal arts college like I'm at is that it's sometimes difficult to get to see really cool seminars and get together and get excited about what we study. So I think this is valuable for everyone, not just you young whippersnappers. You don't have to get off my lawn or my microbial mat. I would like to learn, and that's why I'm here.
That's great. That was well said, Mark. Thank you very much.
And I just want to echo what Mark said, simply because I'm also at a small institution, and we don't have a very large microbiology group. And so it's great to just interact with other people and other people who are other microbiologists who are interested, and come in contact and chat about exciting new advances in our field. And so with that, let's start chatting about the paper. And so at first, we're going to go through a brief outline of the paper. So Laura, would you like to start us off and chatting or giving us an outline of what the paper is about?
Sure. So this is really interesting because it's one of those Nature papers that, I think they had the genome sequence of their organism as a supplementary data. So there is a lot in this paper, and the primary thing that they have presented in this article, given the limitations of Nature, is the identification and characterization of a new antibiotic compound they're calling teixobactin, I think. I listened to Kim Lewis's interview with Nature's Marian Turner that was included in the paper, so I think teixobactin is the correct way to pronounce it, but if anyone disputes that, feel free to correct me. So the interesting thing about this is that they discovered this new compound in a screen of otherwise uncultivable soil bacteria. So they described this iChip, which is a small device that allows them to isolate and then culture in situ individual soil bacteria. And this, to me, is one of the amazing things that's coming out of this paper, is the opportunity to kind of address the 99% of microbial species that we think we are there but that we can't culture. And in doing this, they came up with this antibiotic compound, teixobactin. And so I think that they go through and kind of characterize this, and I think that that's important in and of itself. But the flashiest thing that they did was claim that teixobactin is almost evolution proof, which is what Dave Baltrust said gave him like hermit hands, because that's a very, very extraordinary claim, and it goes against a lot of what we accept as being the general tendencies of bacteria to quickly develop resistance to antibiotics.
Well, I think for one thing, I mean, I don't think this will be proof against persister cells.
Yeah.
I mean, if nothing else. And I think one of the, I mean, even though I want to talk about the antibiotics, I just want to emphasize that this whole idea that no microbe is an island, that they actually work in context with others. I have students isolate codependent microbes from nature as an exercise, so it's easy to do. It's not just about siderophores. There's a lot of interesting work, again, coming from Epstein and Lewis about this. So it's one of those paradigm shifts we need to get to.
Yeah, and so I'm curious. I made a note in there where it said the deal with the iChip is that you kind of, the way they've conceptualized it, if I understand it right, you end up with individual cells in these channels.
Yeah.
What's your thought then, Mark and Karen, about, do you think if this bacterium needs a codependent, it's codependent on another bacterium like you mentioned, if it needs a partner, is that going to be able to, I suppose it depends on how intimate that connection is. So you're probably going to get some things, but if there are certain microbes that need to be close to each other to exchange like compounds that they're producing, maybe you're not going to get those.
Yeah, there are lots that exchange redox mediators, for lack of a better term. Classic example are siderophores, and you can really tell when you get those because you put some extra iron in the plate and they grow there. It's actually a really cool experiment to do with students, which I know you're going to be doing, right? And it's fun to do, but I think you're right. I also wonder, there is some evidence that microbes physically have to touch sometimes.
Right. And
so I think that this is one of those, gambles. This is something that works, and this has been followed up from their previous work, as you don't no doubt are sure to have seen, where they had what were essentially tiny petri dishes that they buried in the aquarium tank, and this is exactly the same thing, but made for a more high throughput approach.
Yeah. Yeah, it's an amazing. I looked at the diagram, the kind of the schematics, I guess. It's a really neat and very tidy kind of way to get at these otherwise unculturable.
So then you think, what about the size of the semi-permeable membrane, right? Especially when you think about some of the evidence that nanowires that connect organisms can actually be extensions of the cell membrane. This is a fascinating idea. But of course, they want to go down to the dollars and cents of new antibiotics, and Lord knows we all need them.
Right, that's for sure.
So then in terms of accessibility of these compounds, it's just through the semi-permeable membrane. So they're still, despite the fact that they're sticking them back into the soil, they're still missing a huge number, even though through the high-put screen they're able to screen some, but in a sense, their system is still limited compared to what in terms of culturing unculturable organisms. Do you both think so?
Oh, absolutely. The hand of Darwin is on all things. And so what I think you're doing here is selecting for particular associations. It wouldn't be at all surprising. But I don't know how else to go about it.
Yeah.
This is a nice approach. It's kind of a nice step beyond the idea of these little petri dishes that they actually buried in the sediment. And that work was done almost 10 years ago now. So this is a good follow-up. Reminds me very much of how fluorescent cell sorting really revolutionized looking at what individual cells can do.
Yeah, sure.
Okay, so that's the first that we...
Oh, I missed her.
Oh, Karen, you've muted yourself somehow.
Sorry, I must have hit spacebar, I think.
Is the space? Oh, well, now we all know. That's dangerous.
Is it? I'm just gonna press it once.
Yeah.
Yes, spacebar mutes the microphone.
Good to know.
Okay, so
wow.
then in Figure 1, they talk about the structure of teixobactin. Is that the correct way to say it?
I think it's teixo or.
Teixo?
Yeah, but.
Okay.
I think that might be for people who are more dogmatic about pronunciations. I think we can get away with both.
So, I guess on an interesting side note, what did they mention in the talk that you were listening to? Were they interviewing the authors?
Yeah, so it was just a clip that was, if you go to the page for the paper on Nature, there's a little clip, and it only takes about 4 minutes. And so it's a very brief interview with Kim Lewis, who's I think the corresponding author, I believe, on this paper. And he talks a little bit about the process of discovering it focuses on the antibiotic, and it talks a little bit about the process of discovering it. And he says some, he really kind of emphasizes the resistance-proof nature of the antibiotic. Probably a little more so. It's a little more extreme of a statement than they make even in the paper. I think they're a little more nuanced in the paper. But he kind of talks up the discovery of the antibiotic.
Okay, I guess so. I know in the past, in my experience with these journal clubs, is there's a total difference between the way that we approach these journal clubs online compared to the way that I'm used to having journal clubs in a seminar room, which is in a seminar room, usually you have somebody who stands up on stage and they present each of the individual figures, and we scrutinize the methods and we go over them. But what I enjoy talking to or having this journal club is that's like we don't do as much of that. Like we do go over the methods, but then you get more of a broad sense and a range of understanding over the actual what's that word that I'm looking for? Not consequences, but impact of the paper over a broader sense. Simply because you have a wider range of scientists who can give comment to those types of things, rather than if you have everybody who's in the same field and then they just scrutinize and say, okay, this person did this wrong, or this person did this wrong because they should have done it this way. So what I guess one of the interesting questions that I had about the results was not so that I'm going and scrutinizing the methods, but I did have a question in terms of what are the normal MIC. So MIC is, if I remember, minimal inhibitory concentration. So what are normal inhibitory concentrations for other antibiotics? Can either of you comment on that in terms of in comparison to teixobactin?
Yeah, so you're kind of typically looking at the microgram per mil. So first I'm trying to think about like with salmonella and their MIC for things for the different antibiotics like ampicillins and so forth. You end up doing these dilutions and you get like 8, 16, 32, and you kind of end up going up on the plate. And it varies. There are some that I've seen where they set so that they set resistance breakpoints based on the MICs, and that's typically set by it's a clinical laboratory standard. So the European Union has a slightly different setting, in some cases, different breakpoints than we do in the States, and they're set by people like the CDC, for instance, will be involved in setting the breakpoints. And some antibiotics have a breakpoint, an MIC, where you consider it to be resistant that's pretty high. Like I think, is it 264? I think is a common one, and then some are a little bit lower. So at like 2, you get what you consider to be resistance. So it kind of depends on the compound, but that's kind of the range that you typically see.
It's funny too, because my students always ask when I do MICs, we do them in tenfold dilutions, and they always ask why when you read why they do them in the multiples that they do.
Yeah.
And I think it comes from 1 to 2 dilutions is where it comes from, I think. But it's an interesting idea. But as you say, there's a big range. And one of the things that always amazed me, taking me back to grad school, is even very similar antibiotics can have very different effects. I mean, kanamycin and neomycin. Kanamycin relatively useless in rhizobium, and neomycin is what you have to use. And there's very little difference between them.
Yeah, so it's interesting because I have to admit that I don't know as much about vancomycin because vancomycin is typically active against gram-positive bacteria since it inhibits the cell wall. So in that case, you'd be, like for instance, when I worked at the USDA, just my previous position, one of the people I worked with worked on Enterococcus. And I think they're thinking about like vancomycin resistance in Enterococcus, and certainly in staph. So they're making comparisons to vancomycin because vancomycin is still, I think, considered one of the last lines of defense against some of these gram-positives. So I could see why they're drawing those parallels.
And I mean, the MICs against Clostridium difficile and Bacillus anthracis are pretty remarkable. I mean, nanograms per mil.
Yeah, so if Karen, if your question was kind of about what scale we're at, the nanograms per mil is quite low. And I think it's interesting too. One of the things as I was reading it, I thought to myself, well, okay, but what about sub-MIC levels? And what we mean by that is kind of like subtherapeutic antibiotic exposure. So we're used to kind of thinking about, well, you have a patient and you dose them with an antibiotic and it achieves this concentration, but really it's a gradient. And it can be a gradient not only in the patient over a temporal or a spatial, but it's also a gradient in terms of the environment because we don't actually, I think with some of these antibiotics, we don't metabolize them. So they go through our bodies and they end up in the environment in their active form. And there was a paper that came out that I cannot remember the authors, but it was a good paper that was examining the effect of subtherapeutic antibiotic concentrations on the maintenance of resistance. So on whether or not populations of bacteria in the environment are maintaining an antibiotic resistance genes and whether or not subtherapeutic doses might have an impact on that. And there's quite a few groups doing really neat experimental evolution, I think, on subtherapeutic doses of antibiotics. So I was waiting to see if they mentioned it, and they did. So they did actually look at subtherapeutic doses, or what they would consider. I guess it's difficult to say what therapeutic would be right now, since we haven't actually developed this into a therapy. But they're looking at sub-MIC levels. And so they're looking to see if you're exposing the bacteria to small amounts of this antibiotic, their landscape is a little different for how it might evolve resistance. And they said that they still didn't find any resistance evolving. So I think that's one of the things that's leading them to making the claim that this is more resistant to resistance than other bacteria or antibiotics.
One of the things that I'm really interested in is what the roles of antibiotics in nature are. And Julian Davies up at UBC has this wonderful idea that a lot of antibiotics are used clinically at much higher concentrations than they occur in nature and are actually a form of signaling molecule. And you know, I always look at antibiotic resistance with that in mind a little bit, because one of the things that people don't think about with building up the cell wall is that it constantly has to be in flux all the time, and except for persisters. And so I just always feel that we're a little stupid when it comes to the small masters. They've got it figured out. They've been working at it a long time. Now, I would love to hear an antibiotic that you can't get resistance to, but I'm real nervous about that. Really nervous. I mean, even simple stuff like saying, look at the structure there. Are you telling me that there isn't some kind of extracellular enzyme that couldn't break that down? It takes you to Van Niel's postulates. Any organism that makes a biological molecule can also break it down. And there are very few exceptions to that. So I'm not saying we should go look for things that break down this antibiotic, but I'll bet you could find it.
So if sorry, Mark, if we go back to a point of bringing up Julian Davies' hypotheses, has he proved that is there proof for his hypothesis, or
yes.
I'm not familiar with this?
I would be delighted to send you a number of papers that I torment my students with. He's found a number of different genes in Salmonella and E. coli that are actually stimulated by subclinical concentrations of antibiotics, and with beautiful pictures of it. It's fascinating work. The idea of a ribosome actually being a kind of a sensory apparatus, which if you think about the way the ribosome works, it is taking in lots of information and changing what it does in response. You know, look at Magic Spot and the effect it has on translation. Same thing. And so this is a very interesting idea. And I'll be happy to send you those papers, which I have in PDF form.
I would love to read them. Sorry, Laura, were you going to say something?
Oh, well, I was just going to say that, yeah, it is. I think that is a very interesting hypothesis, and I think if I am familiar with where it is, I think the outstanding question is, in what circumstances and under what conditions are bacteria producing these to signal? Like, what are they signaling, and what? So I think that we certainly know they have an effect. And then I think, as I understand it, where it is right now is trying to understand under what circumstances would bacteria be using these types of compounds to signal.
And what makes me super nervous is a large number of antibiotics that appear to be produced at the very end of the growth phase out in stationary, which makes me start sniffing, is quorum sensing going on? Is this a group activity? And it may well be true. But as always, the truth's in the middle in a lot of places. I'm not trying to claim that bacteria never use an antimicrobial agent. We know they do. Heck, they use knives, type 6 secretory systems, right? So, this is something that I was thinking about as I was reading this. Are there not 2 targets as well for the action of this antibiotic, or did I misread that?
No, I read it that way as well. I read that there was the 2. Probably going to remember this wrong. It's there's lipid 2, and then there's the precursor also, isn't there? Or am I getting it wrong?
I think you're right.
So I was wondering, Mark, if you can comment on the biosynthesis pathway of the cell wall, because I was trying to look this up. I dug out my undergraduate microbiology textbook, and I could find that there were certain things that I could see, but yeah, if you could comment on that for the benefit of our YouTube viewers, that would be great.
Oh, heck. I mean, there's a beautiful ballet of different carriers. I mean, when you say, what is this? It's like the joke I make in class, what's the molecular weight of starch? It doesn't mean anything to say that. But when you look at peptidoglycan, and I call it bacterial pantyhose because students never forget it when I put it that way, right? And what's funny is it has to be assembled and put into place, so there's a shuttle, and it's the one I can't pronounce, undecaprenol, I think, that keeps shuttling it back and forth. Now, since I love me some gram-negatives, it's even more complicated for them, but gram-positives just have that super thick layer they have to assemble. And I don't want to be of any disrespect to the wonderful scientists that have hammered their head against that problem for decades. It is certainly complex and is a delicate molecular ballet. So with that in mind, you might be able to imagine throwing a monkey wrench in there would be very helpful. But one thing that I keep in mind is there are many instances of organisms that, and I can't pronounce this correctly, autolyse, or there's probably a more elegant way of putting it. And that has to do with enzymes that are kind of expressed when they shouldn't, and that actually causes kind of an over breakage of some of the peptidoglycan, which you need to have during growth. So, I mean, it needs to constantly be doing that. I don't think they're terribly static. Persisters are I don't even think they're a big percentage of what goes on with microbes. But I think they constantly have to kind of resurface themselves, for lack of a better term.
That's my understanding, too. My understanding is that the cell wall is constantly being components are being replaced.
And shed. In fact, there's lots of evidence that those are signal molecules, coming back to that. I mean, isn't that Sportitella pertussis? That's TCT, and it's the same molecule, a segment of peptidoglycan that signals the light organ development in squid. Yeah.
Oh, sorry. Before we move on, Mark, you keep mentioning persisters. In terms of persisters, what are the difference between persister cell walls and normal cell walls?
Persisters are cells that actively become quiescent. That's kind of silly, but they stop growing, and a lot of cell wall targeting antibiotics are relatively useless against persisters. They wait it out. They wait out the actions. They're not actually growing anymore. They're staying quiescent, and that gets them past it. And I mean, that's one of the things that makes me a little bit nervous about MICs and tubes, because we're kind of different than tubes. You can make a joke that we are nothing but tubes, but we're not 2059 tubes.
But you know what? I'm sorry to interrupt you, but I would I would make a comment from my own personal experience that you can actually see, I mean, one of the things that came out of the project that I was working on at the USDA is how difficult it is to actually read MICs, because you do a dilution plate, and I think people conceptualize it as, you know, when you tell someone you've got this dilution plate and you look for growth, they're like, oh, okay, well, one well you don't have any growth, and then one well you got tons of growth, and it's simple. And it's not, as I'm sure, Mark, you know, it's not nearly that straightforward. And so I think you can kind of see how difficult. I have always wondered if you're seeing persisters, or if you're, you know, some of that is just is activity of the antibiotic. But I've always wondered if you might be able to see some persisters in some of those wells. I don't know.
Yeah, I'm almost certain I've seen people do GFP work with that, where they can see them present in using live dead dyes.
Okay.
I'm almost certain I've seen it, but I can't tell you where. And I don't know what numbers they are, but they're a small number. But let's face facts. I mean, microbes are all about the numbers.
Yeah. And
right?
and one of the interesting things that it's been a long time since I've read up on the persisters, because this is actually something that Kim Lewis was involved. And I don't remember which ASM conference it was. It might have been my first, and it's like a while ago now, over 10 years. God, that's frightening. And I saw him give a talk on persisters, and I thought, oh, that's so cool. And I think at the time there were people who were kind of doing the, I don't really know. And he's kind of, you know, continually gathered evidence. I've been trying to follow that story because I found it so fascinating. I haven't kept up with it quite as much, but I think if I remember, if this is still true, that what they'll find is out of a population, a percentage of the cells will act as persisters. Yep. But if you take those cells and put them into a new medium, they're not all persisters.
That's right.
And I thought, what?
And Laura, what does that sound like to you?
That sounds like...
Yes, it does. It sounds like our friend in common, doesn't it?
Yeah.
And we should talk about that sometime, but yeah. I do think that's involved. And there's also this business, and this is because I come at things in a prejudicial way. I think like a geneticist, and biochemists and geneticists sometimes have trouble because of the way that they look at problems. But as a geneticist, we want to believe that every cell in that tube is genetically uniform, and it's just not true. Mm-hmm. And so that's the other part that has to be factored into all this, is how fluid and dynamic and plastic microbes are. I mean, good Lord, it's a microbial planet, right?
Yeah.
Yeah. That's why I kind of laugh when I look at tubes for MICs, because that's going to be very different in the body.
Yeah, absolutely. And I mean, I think for the paper, that's a major point, is which they acknowledge, which is what's going to happen if we try to develop this teixobactin compound into a therapeutic compound. And I think this was where a number of people who were asked to comment kind of made their cautionary statements to say, well, this is very promising, but we honestly don't know yet. The mechanism of delivery, so forth. There's a lot to work out still.
Oh yeah. I mean, I honestly think that, if I know Kim Lewis's style, his research style, the way I do, despite your comment about many years that you've experienced, oh dear God, don't get me started. Oh Lord. Anyway, he's very much a perfectionist about his work, and he's very methodical and straightforward about what he does. And I would not be at all surprised if he's screening samples to look for things that can inactivate that compound. And again, extracellularly, the way that, say, ampicillin or penicillin is broken down by beta-lactamases, that might be the way that that's done. But in the sense that you won't get a mutation to target independence, that might be true. I can't speak to that. But it has to actually reach the machinery. And if you can break it down before it reaches the machinery, that would help make things resistant. So, well, from the first Jurassic Park, speaking of old, there's a wonderful statement that Jeff Goldblum's character made that you remember. I always get a kick out of him because he had an unfortunate excess of personality, something that I can kind of relate to. But the interesting part is that he said, nature finds a way. And that isn't like some homily that you see on a Hallmark card. It's really true. There's so many microbes, so much stuff going on all the time. that I find it very hard to believe there isn't some scam, microbial scam, to break down this compound. Because I don't have the equivalent for nature red in tooth and claw.
Mm-hmm.
You know, pilus and flagellum, I don't know what to say. But that is what we're dealing with here. Anyway,
so now,
sermonizing, sorry.
sorry. So now that we moved on to the elephant in the room, which is, does it really, can it really prevent can resistance not develop with this antibiotic, which is what the authors claim? So we talked about before that it has two targets, and one is the lipid, and sorry, one was the lipid and the other was...
Yeah, sorry, I went and looked that up because it's right in the abstract, so it's really a shame that I didn't remember it. My brain's a little off balance today, but it's so lipid 2 and lipid 3. It binds to motifs in both of those, and those are precursors to cell wall components. Peptidoglycan lipid 2 is a precursor to peptidoglycan. Lipid 3 is a precursor to cell wall teichoic acid. So it's two different cell wall
targets.
Two targets, yep.
So I don't remember if I was reading it in the paper, but was it that the binding target was a sugar? Was that in the paper? No, maybe I wasn't reading it in the paper. Does that sound familiar to anybody?
Yeah.
I don't know what the specific. I didn't look. I think the specific motif. I'm trying to see if it mentions it, but there's a conserved motif that it's supposed to bind to in these lipids.
Yes. Here's the sentence. The target of teixobactin, the pyrophosphate sugar moiety of these molecules, is highly conserved among eubacteria.
There we go.
And that's because, let's face it, I mean, it's sugary sweet, the whole microbe. So that's what we're dealing with.
So then, binding to the claim was binding to the sugar because it's not an enzyme that readily mutates. So then, binding to the sugar would delay resistance. Do you agree?
the long silence. Everyone's like,
what about you, Laura? Do you agree?
I think this compound is extremely promising, but I think, and I have to admit that I didn't have a chance to look at in detail at the methods for how they tried to see if they could find de novo mutations. They did look for this, and I believe they said they went out 27 days.
Yeah.
And I was trying to kind of calculate how many generations that would be. So for experimental evolution, which is what this would fall under, what they are doing is they are attempting to say, if we expose bacteria to this antibiotic compound, and then we grow it out for numerous generations, will we see the development of de novo mutations? So this totally discounts any horizontal gene transfer at this stage. I mean, what we're looking at right now is, are there de novo mutations? Are there new mutations that will arise that will make the bacterium resistant? And they didn't see any, but 27 days, how long has Rich Lenski been going? Years.
Decades, many decades.
27 days might not be long enough. It's hard to know because it depends on the mutational space or the kind of landscape of genotypes that are available in the population as a whole.
Yes.
So what we're looking at is one specific strain, which I can assume is clonal. And so that's one genotype that we're looking to see if it's robust enough, or it has access to the mutational pathways in order to develop resistance. And in this case, this particular genotype didn't. But that's not to say that there's not a genotype out there somewhere that starts at a different, we're starting at different places on the landscape for different genotypes. And so it could be that a different strain has access to different mutations.
And I hesitate to say this because, Dave Baltris with a little child, he gets touchy about things recently, and I can understand because I used to be minus on sleep too. But when you look at RIF resistance in bacteria, there's a trade-off. I mean, you can change the shape of the target so rifampicin can't bind, but there's a fitness cost.
Right.
to the organism. And I could make the same kind of game space argument about anything like this. You mentioned what if we change kind of the arcing between the sugars and the carrier molecules so it no longer fits as well sterically. Sure, we can come up with that, but there'd be a fitness cost too.
Yeah. Yeah, and I think your point is very well made. There's evolutionary trade-offs, which is the bread and butter of quite a few really great researchers, is trying to understand those trade-offs. And some people, and this is how some people work on developing new ways to control bacteria, is trying to force evolutionary trade-offs.
Now, I noticed that they actually say at the end that it's possible that some modification, they don't say degradation, but modification of the antibiotic could happen, and they say, but that's not that didn't happen so well for vancomycin. And, you know, I understand where they're going. The mode of action is similar, but, you know, don't know for sure.
Yeah.
And of course, the more of this stuff we use, the more opportunities. So again, speaking of trade-offs, but I think it's a fabulous new, and it looks like it's not terrifically toxic as well.
Yeah, I'm not as good with those data. I don't know, Karen, is that more your specialty?
Well, I did infect mice, or I do infect mice. So yeah,
I want to rephrase
that. So then the data that they show with the in vivo stuff, they do show that in terms of the amount of antibiotic per kilogram of the mass of the mouse is there's a decrease in the bacteria, so a lower amount of CFU within the mouse. And so there's normally what in these graphs what you would have is you would have a detection limit, which I think they kind of tried to show here at the bottom of the scale. But what we normally do is we draw a dotted line across the bottom of the graph to indicate that that's below our detection limit. And so where they showed, they have here CFUs about 3,000, so they have about 3,000 in terms of per kilogram per lung, and that's not a lot because that's 3,000 total bacteria per an entire lung, and that the control is the amoxicillin. And so based on the data, there is a significant decrease in the amount of CFUs, so it is rather effective in terms of the antibiotic, by what I see. Yes.
Well, I think and we were just talking about this on Twitter, there's this whole business of being too enthusiastic about things. And I think it is good to be really careful about the comments that you make. I mean, I know people, for example, in reading this paper, who will be all upset about the concept of things that can't grow in nature. You know, there's this whole argument you just have to find the right recipe and all the rest. And okay, fine. You know, I'm with you on that too. But this lab has been really wonderful at finding organisms that don't grow except in coordination with others. They have a compound that looks like it has real therapeutic value. Even if something can become resistant to it or create some modifying or degrading enzyme, how is that a bad thing necessarily given the fact that we're running out? Right? This is an approach, a new way to look for antibiotics.
Yeah, yeah. To follow up on that point, which I think is a very good one, to kind of give some context to the broader purpose of discovering this antibiotic and looking for new antibiotics. But one of the other conversations that came out after this was concern about the fact that one of the main things that we need to do in order to address the rise in antibiotic resistance is to start examining the ways that we use antibiotics.
Yes.
And this can become controversial depending on what uses of antibiotics you choose to focus on. Antibiotics are used in agriculture, they're used in clinic, they're used in vet. I mean, they're used a lot. And so it can get kind of testy when people discuss, like, is agricultural usage more or less responsible?
Yeah.
You know, I'm not sure that that's a point that needs to be discussed and the data need to be collected, but on a broader level, I think one of the things that came out of this was people being concerned that those efforts to curtail our antibiotic use or to at least be more judicious in our antibiotic use might get overwhelmed by this idea that, oh, finally, we'll just discover new ones and we'll be great. You know, and so people were saying we can't lose sight of the fact that even though this is promising and even though this looks like it could help us, we can't lose sight of the fact that we have to try to do something to be more careful and judicious about how we're using antibiotics, including this one if it makes it to market. So,
in a way, it reminds me of the argument about Roundup, Roundup ready plants, and the criticism is it just encourages people to use more herbicide, and I appreciate that style of argument.
Yeah. What were you going to say, Karen?
Oh, I was just going to say that because the amount of time that it takes to develop the antibiotic is anywhere between 5 to 10 years.
Absolutely.
So, we're reading this paper right now, but the likelihood of seeing it on the market is probably not for a long time. So I totally agree that in that period of time, before we see these new antibiotics come to market, we really do have to be careful with the amount that we have in our hands, because those are the only tools that we have currently right now. So it's either we have 5 to 10 years to prevent resistance. Like it's essentially our timeline. Or else then we have to wait for the next weapon, because it's either so it's like 5 to 10 years, and then that's time's up, and then you'll be able to get the next one. But in the meantime, these are the only tools that we have, and we need to use them judicially.
Yeah, absolutely. I agree.
Okay, so it's about 12:06. We started late today.
We did, yeah. We had some technical difficulties. Thanks for being patient while I tried to figure it out.
No, thank you for setting up. So I just realized that I didn't end up having everybody introduce themselves at the beginning, so I introduced myself. So I want to quickly at the end. We start with Laura. So Laura, you can introduce yourself, or where you're from and what kind of research that you're doing.
Sure, absolutely. So I'm Laura Williams, and as of January 1st, I'm a new assistant professor in the biology department at Providence College in Providence, Rhode Island. Previous to that, I was working at the USDA on a project to look at genomics in salmonella and specifically antibiotic resistance. So this paper is very interesting to me in terms of the antibiotic resistance aspect. And I am hopefully, I've got some new student researchers, 5 students working with me this semester, and we're going to be starting to examine predatory bacteria, which is near and dear to the heart of Mark, our co-attendee of our journal club here. So he'll probably tell you a little bit more about that.
Oh no! All right. Well, I'm the entropically challenged member of this dynamic trio, or terrific trio, if you'd like. I'm Mark Martin, and I'm an associate professor, the oldest associate professor in the world, at the University of Puget Sound in lovely Tacoma, Washington. And I spend most of my time teaching, as does everyone at a small liberal arts college, but I do have a small research lab, and we investigate predatory bacteria and symbiotic associations. Delavibrio is the faithless mistress that loves me sometimes and not others, as Laura, I hate to tell her, will discover.
Yeah.
So buy Delavibrio lots of flowers, Laura. It seems to help. And then there are some other predators that I'm looking at as well, and I've become increasingly interested in tardigrades and their microbiota and things of that nature. But we have a lot of fun doing research. It's not like a big school, but we're having a grand time, and I'm delighted to have collaborators and people who are kind to me, like Laura.
Collaborators are always good.
Definitely. Also, if you're interested in either Laura or Mark's research, or if you just want to chat with us or find them, we are all available on Twitter. You can also go to the G+ Micro website, the Journal Club website, and you can find, we'll be posting more things about the Journal Club. We'll post future Journal Clubs, and if you have comments or other papers that you suggest that you want us to discuss, we'd love to hear from you. And so that's all for us today. And so thank you for joining us. And this is us signing out. Bye. Bye.